Maybe Dr. Vamsi, we can reduce the music and we can start discussing on few things. Dr. Vamsi, can you hear me?
Yes, yes. So,
Okay.
welcome participants. We will be starting the session very shortly. Yes, sir. We'll be starting
And,
formally. We'll be starting the session very shortly. Yes, sir. You
yeah.
can go ahead.
Well, greetings to everyone from all over the world, wherever you are joining us from. As Dr. Vamsi Dasu mentioned, we'll be starting in few minutes. And while we are waiting and to start, we would like to talk about the institution of the panelists. For example, Dr. Kachu was talking about his institute. So, her institute, sorry. Dr. Kachu, can you kind of you were saying the what type of projects you were doing, for example cost effectiveness projects, and you were mentioning about your work versus Dr. Edwin Michael's work. He presented in the yesterday actually. So what were you mentioning?
Yes. So, good evening and good morning. I don't know which time zones we are in for others. So, good evening and good morning, everyone. Thank you. I would like to start with a thank you note. Thank you for inviting me in this wonderful conference, which is actually very well organized event, I must say. And I would also like to congratulate organizers, for thinking and coming up with such a brilliant topic for the conference. Actually, this is a need of the hour when we are struggling with pandemic and we are trying to establish evidence-based practices, and health economics is playing a wonderful role in that. That's how we are contributing in the development of the entire healthcare ecosystem of the country by doing evidence-based medicine and evidence-based practice, and we're spreading that across to the country. So it's like the basic gap these days in the entire scenario which we found in our country was research translation. As I will again go back to Dr. Michael's yesterday's talk, he talked that your research question should be ideal to arrive at some conclusion, and
Mhm.
that research translation is not happening. There isn't a synchronous relationship between researchers and industries. So that is what we are doing. So our institution, our ecosystem is a bridging gap between industry and academia.
Thank you very much. I mean, this is what exactly why we decided to have this type of panel where we have Dr. Ramachandran is from Drug Development Institution. Dr. Brian Val is a public health epidemiologist, and Dr. Wu and Dr. Mac Hyman are pioneer in mathematical modeling arena. And many times we are on own discipline and we focus in our own way of in our own lenses, and sometimes we try to do the modeling interdisciplinary, for example, but we don't understand the terminology. I mean, it's very simple, like the word model for mathematician is an equations, but for a social scientist it's a flow chart, and for a biologist it's a mouse. And so the same word sometimes we use it, and it's very quite drastically between the fields actually. So thank you for giving. And health economics is something that I'm now seeing it, because now I'm working with an institute also as health economics. Like we model the trends, patterns and trends using mathematical models. On the top of it is a cost associated with it. So I think that part we don't do it very often. And that is probably your one of your areas of your
Yes.
institute. um,
Yes.
Yeah, Dr.
Maybe.
Anuj, sorry to interrupt you. Maybe I can introduce the moderators for this session, and we can
Sure.
informally continue the session till the last
Sure.
panelist, the other panelists
Sure.
join. Okay.
Sure.
So now let us formally start, and of course we wait for the panelists to join. So we welcome you all, the participants and the panelists and the moderators for the most awaited evening session of the Indo-US conference. For this evening session, we have a panel discussion titled "The Hard Lessons from Modeling COVID-19 Dynamics and Health Policies." On behalf of the organizing committee, Dr. Anuj Mubai and myself, we welcome the panelists and moderators once again for the panel discussion. So we have two moderators moderating this panel discussion. The first moderator is Dr. Anuj Mubai. He is currently Associate Director in the Advanced Modeling Group of Precision here and Distinguished IBA Fellow at the Center for Collaborative Studies in Mathematical Biology, Illinois State University. Dr. Anuj is a former faculty at the Arizona State University, where he directed Presidential Award-winning Mathematical Theoretical Biology Institute (MTPI). He is an applied and computational mathematical scientist with more than 10 years of experience in disease modeling and health decision analysis. His research is driven by quantitative and qualitative modeling of problems of interest to the public health and social sciences. His recent research interests include the development of new cost-effective tools for getting the impact of infectious diseases while increasing the understanding of mechanisms that drive their prevalence and mitigating morbidity-related consequences. The next moderator is Dr. Bk Birthwatch. He's from as a Sathya Institute of Higher Learning. He's currently assistant professor at the Department of Mathematics and Computer Science. He graduated from Sathya Institute of Higher Learning with an MSc degree in 2006. He also did his MPhil from the same institute before going on to do his doctoral research work. He is interested in his doctoral research work focused on qualitative study of nonlinear ordinary differential equations. He worked mainly on the qualitative theory. Presently, he is interested in working on application of analytical techniques to the study of solutions of different types of ordinary differential equations. Thank you. Welcome you all. Over to the moderators, Dr. Anuj Mubai and Dr. Bk Birthwatch. Thank you. Once again, I thank all the panelists. Thank you.
Thank you, Dr. Vamsidasu. Maybe we can start. Dr. Bhardwaj, are you there?
Yes, Dr. Anuj, I'm here. Thank you, Vamsi, for your introduction.
Yeah, so maybe we can start the panel discussion, and it's already 10 minutes past, 15 minutes past. So, and as we joined by other panelists, we can continue for addressing some of the things related to the other panelists. So maybe we can start with the slides. So Dr. Vamsidasu did mention about our background. So I welcome everyone formally now on behalf of Dr. Bhardwaj, Dr. Vamsidasu, and others who are behind the scenes in organizing these things. Just want to add that my background has been, my training has been in applied mathematics. But I have also had a clinical experience in Cleveland Clinic, Ohio, before I kind of had a faculty position for a number of years in Arizona State University, before actually current position in as a health economist modeler in Precision HUR. So that's the way. Dr. Bhardwaj, would you like to add something else, or your description was okay?
Yeah, hello everybody. Sairam. On behalf of Department of Mathematics and Computer Science, Sri Sathya Sai Institute of Higher Learning, we welcome everybody for this panel discussion. I'm Dr. Bhardwaj, and we are very happy to host this conference. And then our students and all other participants of the conference are very excited for this panel discussion. Dr. Anuj, over to you.
Okay. Can we have a slides, please? And I'm not sure if Dr. Brian Val is still there. Okay. Okay. So next slide, please. So I want to dedicate this, actually we dedicated the whole conference, including the panel discussion, to Professor Fred Breuer. He passed away recently, I mean, actually a few weeks ago. He was my academic grandfather and my advisor's advisor. but he is a well-known figure in applied mathematics, mathematical modeling. He has a tremendous amount of work in mathematical epidemiology, um, and his books are widely very popular in the community. He was a faculty of University of Wisconsin Medicine in a math department for 36 years. Um, but last a couple of decades, he was a professor at emeritus at University of British Columbia, Vancouver. So
Just
next
wanted to
slide,
add,
please.
yeah, Professor Brauer is as close to the analysts as he is to the applied mathematicians. So just wanted to add the angle of being an analyst to Brauer,
Yeah,
Professor Brauer.
yeah. Okay, thank you.
Yeah.
Next slide, please. So, what is the focus here? The focus of is today's panel discussion is the challenges we have seen in COVID-19 pandemic. It's not only medical challenges, public health challenges, but if there is a problems in the community, there is all this whole supply chain issue. From modelers are also equally was realizing that they were unprepared in understanding many aspects of the COVID, and that is why this particular panel discussion is carefully crafted with experts from different areas, not in only in academia and industry, but also from different fields. Um, and so the challenge today, the discussion we would like to have today is what were the challenges associated with existing modeling techniques that we used. Why were we slow in adaptiveness to the data collection process and linking it to the models? And what was the challenges associated with forecasting techniques? And eventually, why we talk about these all things is because we want to link it to the decision making. What was the gaps in the decision making? Eventually, we catch up to a lot of things, but initially we were struggling to understand many different aspects of it. Um, and then also want to talk from the student's perspective, the shifting patterns of professional opportunities. It is a sudden shock in the system in many different ways, not only in the health aspects but also in educational system. We are now teaching online most of the things. So what is the shifting trends in professional opportunities, jobs for examples, or teaching mentoring methods? If you can shed lights from your work that you have been doing, I'm addressing two panelists here. That would be awesome, actually, if we could talk about that. And so these are the main ideas that behind why we kind of constructed this panel discussion. Next slide, please. So I want to acknowledge, first of all, Dr. Anamika Mubai. She is a PhD in medicinal chemistry, and she helped actually in organization of this panel discussion, especially the questions that we finally decided to discuss about, which was a very difficult task and a lot of people. And Dr. Vamsi Dasu, he's also a co-organizer of the conference, and he has a PhD in mathematics from Sathya Institute of Higher Learning. Next slide, please.
We will introduce our panelists for today. We will first introduce the panelists who are with us right now. Dr. Kavitha Kachoru. We have Dr. Kavitha Kachoru with us. Dr. Kachoru is the head of the Department of Health Economics and Outcomes Research Division at Kalam Institute of Health Technology, a project of Department of Biotechnology, Government of India. She has 12 years of experience in health economics. Kalam Institute of Health Technology is a Government of India funded health technology policy institute which aims to facilitate focused research on critical components pertaining to medical devices and drugs by supporting institutions involved with R&D, industry, policymakers, and knowledge repositories. Dr. Kavitha will provide background on her research interests on health technology assessments. We then have Dr. Ravishankar Ramchandran. Dr. Ravi is the chief scientist, molecular and structural biology, sophisticated analytical instrument facility, CSIR, Central Drug Research Institute, Uttar Pradesh, India. His work represents fundamental contributions to the disease biology of TB and in the development of new therapeutic approaches against antimicrobial resistance. Dr. Ravishankar Ramchandran will provide background on his research interests on the disease biology and in the development of new therapeutic approaches. We then have Dr. Brian Val. Dr. Val works at the Johns Hopkins University, USA. He is an epidemiologist with a broad interest in the relationship between health systems and infectious diseases in low and middle income countries. His research focuses on understanding the changing epidemiology of pediatric experience, the pediatric respiratory diseases in the context of new health interventions. He has experience contributing to field studies and disease burden transmission modeling projects. Dr. Brian will provide background on his research on global disease epidemiology, health systems, and public policies. We also have Dr. John Hong Wu with us. Dr. Wu works at the York University, Canada. Dr. Wu is an applied mathematician and the founding director of the Laboratory for Industrial and Applied Mathematics at York University. He holds the lifetime title of University Distinguished Research Professor and has been a senior Canada Research Chair in Industrial and Applied Mathematics at York University since 2001. Professor Wu is the first recipient of the Canadian Applied and Industrial Mathematical Society's Research Prize for his very significant contributions in the area of infinite dimensional differential equations with applications to neural networks and population dynamics. He is recognized for his expertise and contribution in the fields of nonlinear dynamics and delay differential equations, neural networks and pattern recognition, mathematical ecology and epidemiology, and big data analytics. Dr. Wu will review advanced modeling techniques in mathematical epidemiology, and in particular, his collaborative research with public health professionals in government and industry. Next slide, please. So, coming to the schedule of this panel discussion, we will begin with each of the panelists spending at least a maximum of 5 minutes on their research focus. And then we have the round where we have one question to each of the panelists. And this question will be answered by this particular panelist to whom the question is posed. And then we elicit responses from other panelists too. After which, if time permits, we can have some questions from the audience. After the questions from the audience, we will have the closing remarks by the moderators. Over to you, Anuj. Dr. Anuj, I think you are in mute.
Oh, yeah. So we would like to invite our first panelist, Dr. Kavitha Kachoru, to kindly share her research focus, and please join us in welcoming her, Dr. Kachoru.
Thank you so much for inviting me. I would like to take you through what all we are doing at my institute. So I would like to share some presentation which I have prepared for all of you. So how do I do that? Where is the share button?
It will be on the bottom of the
Yeah.
window browser.
Ma'am, you already
It's
shared the
not.
screen. You have to get the slides on the screen, ma'am. You already shared the screen.
Okay,
Please,
okay, that's why. Yeah. So my slides are visible?
no, ma'am. You have to keep the slides on. It's not visible.
Okay. Let me try again.
Yeah, now it is coming.
Not coming.
So you have to select the window option there in the browser itself.
Okay.
We were seeing your desktop, but now we are not seeing it.
Okay. Give me a second. I'll stop sharing and then again share.
Okay.
Maybe we can start with the next panelist to save the time with this technical glitch.
Sure, no worries. Maybe we can invite. I would like to request Dr. Ramachandran to kindly share share his research focus, and please join me in welcoming Dr. Ravishankar Ramchandran. Dr. Ramchandran.
A very hearty welcome to everybody. This is a wonderful opportunity to meet like-minded persons. I'm a hardcore researcher in drug discovery, and then the COVID pandemic situation pushed me out of my traditional boundary of focused research into drug targets, assays, biochemistry, structural biology, and so on, and got me into a multidimensional thing in the public health scenario, especially liaising. I also happened to be on the Prime Minister's advisory council, the committee constituted by the PSO, the Principal Scientific thing to the Prime Minister, for drug repurposing, among other things. And in the recent past, I've been extensively liaising with the state government on various aspects to counter the pandemic. So I'll just share a slide just to show you some of the thing. Just a second. Hmm. Is it visible?
Not yet.
Okay. I initiated the sharing.
So after sharing,
Okay, I don't need the slides. You see, I don't need these slides also. I will just tell you right away. Hope it works well. Share. And here we are. Does it share?
no, not yet.
No.
You have
Yes,
probably
it's coming
Yes.
up.
Oh, great.
yes. Now it's coming. Yes.
What should I?
Yeah, we're able to see your slides.
Oh, wonderful. So, this is a summary of some of the recent work we have done in the area of countering the pandemic. This is my beautiful institute, the Central Drug Research Institute at Lucknow, India. And it's on a sprawling 61-acre campus. And I'm looking forward to welcoming some of my fellow panelists also here in the near future. So, when the pandemic started, CSAR as a whole and our own institute as part of CSAR started work on several fronts. The most important thing that was demanded was testing patients, the RT-PCR test for patients. There was only a couple of testing labs, and today now we do more than 1 lakh tests per day in the state of Uttar Pradesh. And our own contribution has been within a month, we developed a diagnostic testing facility for COVID with all the protocols. And today we have screened more than 300,000 patients. And then we also were requested by the state government to sequence the whole viral genome to find out how the disease is spreading, which variants are of concern, etc. Both in the first wave and in the second wave. In India, we have had 2 waves so far in the state of Uttar Pradesh also. And right now, the cases are at an all-time low. It's quite a safe place to be in right now. And what we found was in the second wave, it is largely driven by the Delta variant. And in the first wave, it was the other Alpha variant. And in terms of new drugs and repurposing of drugs, we shortlisted a few molecules. You see, when you are countering a pandemic disease, you don't have time to develop a completely new drug from scratch. You use whatever weapons you have in your arsenal to counter it. And one very important approach there is to use your arsenal of known drugs. So umifunavir is one of those drugs which is a known antiviral, extensively used in Russia, China for the past 20 years for influenza and cold, etc. What we did was very quickly made the process technology, transferred it within 1 month to the industry, and facilitated its manufacture, and then conducted phase 3 clinical trials for umifonavir, which have just been concluded. And we've got fantastic results, and we're going to take it ahead in further things before commercialization. So this is something that we did. We also developed more than 6 APIs, and in the pandemic situation, you can't import. We found that the supply chain and logistics support systems were severely curtailed. So we developed systems to make these APIs using natively available chemicals. And of course, in the initial stages of the pandemic, even hand sanitizers for the police staff, etc., were not available. And so we made a lot of stuff and helped the local government to spread awareness and etc. And these are a few glimpses of the facility set up in a month's time. And I'm sure this kind of a response was done by many, many centers across the country too. And a big salute to all of them. And this kind of summarizes the title of the clinical trials that we carried out, the phase 3 randomized double blind trial, using again in 3 centers. And right now we are hoping that this would be commercialized very soon. And I'll stop here because in 5 minutes this is some of the thing, and I look forward to telling more things as and when the panel discussion progresses. Thank you.
Thank you very much, Dr. Ramachandran. This is amazing to know that thing, because now actually something like Tamiflu or Tammy COVID is about to come, we are hearing in the news. So right, appropriate, and I think we have more questions on your talk,
Yeah.
but we will hold on to those curiosity for later on. Um, Maybe I would invite again Dr. Kachoru to please share her research focus. And it's natural, like our, this is what we are learning in this COVID times. How do we adapt to the changes? Or so there's technical glitch, for example. So please, Dr. Kachoru, the floor is all yours.
Thank you so much for inviting me. Is my slide visible now?
Yes.
Yes, it's visible. Yes.
Okay, so as I have already shared, I'll give you a virtual tour of what we are doing here and how this pandemic was a big opportunity for us to really translate the research into the practice. So what we actually did, we were able to serve to the last mile with the help of research and academia and industry collaboration. So all these slides will give you a glimpse of that. So this is our campus, Andhra Pradesh Medical Technology Zone, India's first integrated medical device manufacturing ecosystem. Saving lives, saving nation. This is our motto. The pandemic had placed unparalleled demands, and we have also provided means to strengthen the healthcare industry with opportunities for innovation and cost efficiencies. AMTZ played its role in helping tide over the crisis by rapid escalation of manufacturing of essential and critical medical equipment. So, in the entire ecosystem of Andhra Pradesh Medical Technology Zone, we have different companies, and one among them is Kalam Institute of Health Technology, which acts as a binding force on all other sub. Companies which are located here. So at Kalam Institute of Health Technology, we do health technology assessments. We have a partnership with Department of Health Research, ICMR, and it's called at HTAIN, and we are a resource hub for that. So what all projects are delegated to us? We do a health technology assessment. We do check clinical efficacy of equipment, medical devices. If it is a new process, if it's a new drug, we evaluate the clinical efficacy, we evaluate its cost effectiveness, we evaluate all the ethical concerns, regulatory aspects, we include societal components also, and present and make a comprehensive report in front of the government, as there are different agencies across the globe which are doing health technology assessment, and which is translating at the end, going to ministry and leading to changes, decision leading to changes at the policy level. So that's what we are doing here at Kalam Institute of Technology. There are subdivisions also in this. So another division is cell for market intelligence and trade, cell for technology transfer, cell for product realization. So in our ecosystem, we have Center for Electromagnetic Capability and Safety Testing, Center for Biomaterial Testing, Center for 3D Printing, Center for Gamma Irradiation, Center for Molding, Center for Lasers, MRI coils, and many other industry service centers. So this was the main contribution of AMTZ to the entire nation, where we were able to produce 1,000 ventilators in a day, 500 oxygen concentrators, 1 lakh N95 masks, 5,000 PPE kits, and 1 million RT-PCR kits. Other products manufactured at AMTZ are superconducting magnets for MRI, infrared thermometers, and many more. So, as we all know, this due to COVID-19, healthcare was put in the limelight, and we were thrown in such a situation to convert this pandemic into an opportunity, and that's what we did. We converted everything, and we were able to serve the nation with all the enhanced in-house production and supporting Make in India campaign. So attending needs through innovation. These are now I'll present some of the disruptive innovations which we came up with during this pandemic. So iLab is an infectious disease diagnostic lab with a biosafety facility capable of RT-PCR, ELISA, and 30 more tests were introduced to ensure ease of testing in rural areas. As I was mentioning to you, the resource translation. So this is one among the industry research partnership, and we were able to deliver services to the last miles. Second is O2 Home. So an app available on Android and iOS platform enables its users to rent oxygen concentrators on per day basis. So as we all know, there was a huge critical shortage on oxygen. We came up with an idea like, when Uber can be made available, why can't we make oxygen concentrator available to anyone and everyone who has their mobile phone app and who can just order Uber? Similarly, we can order oxygen concentrators. So this was an amazingly interesting concept, a disruptive innovation concept which we introduced into the entire ecosystem to help people across. Again, these were two very beautiful initiatives which were planned by us, Oxywheel, with an ability to generate oxygen at the rate of 250 liters per minute. This mobile oxygen generator ensures oxygen generation on demand at any required location. So, oxygen on wheels. Another interesting innovation was Safe Zone, the infectious disease containment and treatment zone, which can be moved anywhere when needed, ensures availability of beds during crucial times. So these safe zones are prefabricated containers on which it's like a ready-made container hospital which can be transported anywhere where there's a shortage of beds, there is a shortage of medical devices, there is a shortage of infrastructure. This is a one-stop solution which we came up with, and we deployed all these solutions across the country wherever there was a demand. So distribution footprint. So this map is explaining where all we have delivered oxygen concentrators, ventilators, diagnostic kits. Even we were able to supply oxygen concentrators to other countries of which are from BRICS nation. Uh, in addition to all these things, we have a council where we even teach human resource, because during the pandemic, coming up with disruptive innovations was not enough until there was a human resource which could parallelly support all these initiatives pan India. So simultaneously, we planned so many online courses to enhance the capability of human resource in terms of their knowledge, and they were able to do minor corrections on these indigenous medical devices on field at any village, at any city. So again, this training in terms of medical devices was very crucial at that time, which we were able to do. So, if I talk about the projects which were completed by us, this is just a glimpse of a few projects which we've completed and which were presented in front of the ministry, and they were approved. And after this, orders were given to different companies who were manufacturing these innovations, and there was a huge paradigm shift. As we all know, this low-cost portable ventilator was the talk of the town during the pandemic, and luckily we were just doing this HTA on low-cost portable ventilator just before the pandemic, and we were about to finish this report just the pandemic started. I really found that this low cost portable ventilator is quite effective, clinically effective, it's cost effective, and it can really save lives. Second interesting innovation which we did a health technology assessment on is Thermolytics based health cancer screening tool. And it was found this tool has amazing sensitivity and specificity, and it is really cost effective, and it can really change the scenario, because mammography is a radiation induced screening tool, maybe a second level tool. First level tool is self breast examination, but this is 100 times better than that. This proved 100 times better than that while doing the analysis. And this is radiation free, sensitivity specificity is quite high compared to mammography. And again, if we bring this innovation in our ecosystem, it can really change the scenario. We can since breast cancer is the second largest cancer due to which deaths are happening, so we can really reduce that if this intervention is introduced to the health system, which is yet to present to the ministry. So another intervention was we did a health technology assessment project on PET for cancer care in India. We were given a task to calculate the cost, to do a budget impact modeling for establishing a PET CT scan center, um, and and a cyclotron center. So we came up with the calculation that 17.08 crores would be required for establishment of PET CT scan facility, and 58.63 crores would be required if we are setting up a new cyclotron facility. And
Dr.
we did
Kachoru,
yes.
that was a beautiful virtual tour of the institute, because
I'm about to
we are
finish.
short of
It's the last
time.
slide.
Yeah, thank
It's
you,
the
thank you,
last
thanks a
slide.
lot. Thanks a lot, thanks a lot.
So we did, lastly we did economic analysis for COVID-19 diagnostic kits. As there were many kits available in the market, we really wanted to find out which is the most cost effective kit. That's what we did, and we recommended this to the government. So all these projects were a game changer. Similarly, these are different projects which really helped out us during the pandemic. So that's it. Thank you so much.
I'm sorry, Dr. Kachoru, I had to interrupt because we are falling short of time.
No,
But
no,
that was a
I'm
wonderful
already
virtual
done.
tour. We are very happy to know that so much is happening around us in India. There is this talk about Atmanirbhar Bharat, and then we are able to see this directly from your presentation. Thank you so much. Dr. Anuj?
Yeah, so I just want to second that thing, and I think the health economics component from a modeling, it's important that this is a discussion actually, and I think this is very, very important as we are going forward with it. We talked about where the modeling um, could be very helpful in these aspects. So, like health economics is quite a good field. Quite, they use a lot of good models for cost effectiveness. We talked about trends, but on top of it, the economics aspects is very important. If we do a very good job in diagnostic test and we cannot, and it's very expensive or it is not cost effective, then it is not good. The cost effective component is very, very key actually. And thank you, Dr. Kachu, for giving this wonderful tool, virtual tool, as well as the projects that you are part of it. So thank you very much once again. um, I would now move on further to and invite Dr. Val to kindly share his research focus and in epidemiology, public health, and he has been doing wonderful thing. I've been following him on social media, so I know what is the different project he's part of, some of it, if not all, and I'm excited to hear more about it. So, Dr. Val.
Thank you, Anuj. And thank you. It's so wonderful to hear all of the fantastic work that's happening in Andhra Pradesh and Uttar Pradesh and elsewhere. And so really, really excited to be with you all today. As Anuj was mentioning, my name is Dr. Brian Val. I'm a faculty member and an infectious disease epidemiologist at the Johns Hopkins Bloomberg School of Public Health. I've been working in India on vaccine-related issues for more than a decade now. Normally, I stay in Delhi, but I'm actually joining everybody from Goa, where I'm actually on holiday at the moment. But it's great to be with all of you nonetheless. I'm excited to talk today with you about some of the work that I've been doing on modeling different vaccination scenarios specific for COVID vaccines in particular, primarily in India. In the context of modeling these different COVID vaccine scenarios, I've been working with several different colleagues at many universities, Harvard University, Ashoka University just outside of Delhi, the National Center for Biological Sciences in Bangalore, and as an American who is and has been living and working in India for as long as I have, and as being part of these collaborative efforts, I'm really excited about this conference and the focus on existing Indo-American collaborations, and also opportunities to expand Indo-American collaborations. So, I'm happy to present some of our work today, and if there are others who are interested in collaborating, I believe our email addresses will be shared at some point during the presentation if they haven't already. I think maybe on the title slide, our our email addresses were listed there. I'll share some slides in just a moment, but I want to kind of just maybe start by reflecting on why and how we use models. And I think the world now has been exposed to models as far back as January and February of 2020. So the world has now been exposed to mathematical modeling for almost more than a year and a half now, if they weren't already previously exposed or involved in mathematical modeling. And we use mathematical models to synthesize evidence and scientific knowledge. And I think that that's a really important perspective on the use of models, because if we think back to the models that we were all digesting or maybe even working on at the very beginning of the pandemic, they were, these very simple compartmental models, SIR models. And the reason why those models were very simple was because, as tools to synthesize evidence and scientific knowledge, the scientific knowledge at the time was quite limited, and so we had to work with what we had available, and that's one of the reasons why you saw in, say, for example, top tier journals, these very compared to the complexity of models that are being published now, these much more simple models. And I'll talk a little bit maybe later about kind of some thinking on how we use models in three ways: understanding how complex systems work, forecasting, and then looking at multiple scenarios to consider different potential options. I kind of think of mathematical models in these three distinct ways. And so if I can share my screen, I'll just quickly run through some of the modeling that we've been doing in India. Just give me 1 second.
Sorry to interrupt. We welcome Dr. Hyman, James
Yes,
Hyman.
I'm sorry, I had a time zone mix-up.
Yeah, we welcome you, sir. Dr. Anuj, you could welcome Dr. Hyman.
Yeah. Greetings, Mac.
Hi,
I'm
Anuj.
glad you could. I know you also moved to a different location. I mean, you moved from New Orleans to Los Alamos. So I know that thing. So I think you are in the process of moving also. So thank you for joining actually in this panel discussion, Mac.
I'm delighted to be here. I apologize for being late. For some reason, I didn't think it was going to start for another 10 minutes. So
That's okay. So I think Dr. Val is giving his little research focus introduction. So maybe we can have after that yours and Dr.
I'm glad to.
Dr. Val.
Dr. Brian, you can
Hi, Professor
share
Hyman.
your slides.
Welcome. Okay. I'm getting an error message saying that Chrome doesn't have permission to capture my screen. I haven't used StreamYard before. Maybe I can just, I'll try one more time and see if this works. Just give me 1 moment. And if this doesn't work, I'll just maybe talk it through.
Look at Mark's lab. It's very, very chaotically ordered.
Are you able to see my screen?
No.
Not yet.
No. Okay. Oh, it still says I don't have permission. So anyway, I'll just talk through kind of what we did. This was work that was commissioned by the World Health Organization, and work that I, together with my collaborators at Ashoka University and the National Center for Biological Sciences in Bangalore, led on looking at different vaccination strategies, and how they can help maximize in-person schooling, and how vaccination strategies can also help in lifting NPIs. And we used the best available evidence that we had at the beginning of 2021 on vaccination effectiveness, especially as we learned more against the effectiveness of the vaccines against emerging variants of concern. integrated that into our models, as well as single dose and two dose efficacy and effectiveness estimates as well, and how well they work against different endpoints, less severe endpoints as well as severe disease and death. We also used seroprevalence data that has been published by the Indian Council of Medical Research, ICMR, at the time looking at post first wave, a seroprevalence of around 22 to 24%, and using network models, looked at different opening up of schools upon reaching vaccination coverage of different levels, together with seroprevalence also at different levels as well. And what we found was that looking at the levels of seroprevalence that we see in India at the moment, post the second wave, including seroprevalence among children, which is relatively high, especially compared to other scenarios. We identified that opening schools at this point would not contribute to a significant increase in cases overall in the country. There would be a modest increase in cases among children potentially, but of course children are at much lower risk of severe outcomes related to COVID. And so given this overall given this overall kind of minimal, if any increase in all in symptomatic cases overall, we kind of concluded that at this time, given the net seroprevalence afforded by both vaccination and prior exposure, that it would be reasonable to begin opening schools again. And of course, we know of all of the secondary impacts of having schools closed, especially on marginalized students who come from, for example, low socioeconomic families. The one caveat that I would mention here is that this is very context specific, given existing immunization coverage, given existing seroprevalence estimates, given network structures. We used India-specific network structures in developing these models, and so I would caution against extrapolating any of the results that we found from this effort to other settings as well. I think for example, in the U.S. or elsewhere, there may need to be a more local approach to considering how and when to open schools. So maybe I'll stop there, and apologies that I wasn't able to get any of my slides up and running, but happy to share some of these slides soon, and we are hoping to have a preprint and hopefully a publication of this work available shortly, and look forward to sharing that with you all as well. Thanks, and look forward to the discussion today.
Thank you, Dr. Val. This is amazing. Actually, this is what we had Dr. Gotham Menon from Ashoka University also talked about. So maybe some of the work that you coincide with him as well.
Yeah, so this is work that we did together, Gotham and I and Sandeep Krishna from NCDS together worked on this project.
Thank you very much for this broad description about what work you have been doing in India as well. So I would like now to welcome Professor Wu. I know him from long time actually, and his work I follow. He has done work in many different areas, but also one of the important thing is he's a senior Canada chair, through which he has been actually working with Public Health Agency of Canada, if I'm not wrong, or the Ontario Health Departments on a regular basis. So he's involved in even the policy making modeling aspect. So please join me in welcoming Dr. Wu. Dr. Wu, floor is yours.
Thank you very much. Greeting from Toronto. And it's been really a privilege to be part of this distinguished panel, including our Mark Hammond, who's joined us now. I am a mathematician. However, I did direct the Health Research Institute, and I'm also directing a advanced disaster, emergency, and rapid simulation center. So I will talk about the research focus of myself and my lab, but I will also touch on some of the research focus of the Research Institute and the National Research Network that I'm directing and leading. So in terms of myself and my research lab, the Laboratory for Industrial and Applied Mathematics, we mainly using dynamic models and bifurcation analysis to conduct scenario analysis, to conduct researches to inform stochastic optimization of resources and outcomes. So we do, as Dr. Mambay mentioned, we do provide nearcasting and forecasting of epidemic trends for different levels of government. For example, we do provide biweekly nearcasting trend for the Ontario government, and we do provide long-range forecast for the federal government of Canada. We use the model to provide assessment of infection risk, to conduct evaluation of different interventions, and our focus is more on optimization against different strain and constraints and against different target, including, for example, optimization in terms of reducing hospitalization, reducing the use of ICU, and not minimizing the impact on social economic activities. So we do that in terms of optimize the reopening procedures, the stabilize reopening, the timing, the scale of reopening. And we do that in terms of vaccine, for example, the dose interval, what would be the optimal interval in the consideration that initially the vaccine supply is limited. So what would be the optimal interval between the first and second dose in order to maximize the impact of vaccine in providing the herd immunity in a timely fashion. Of course, we're now working on the third dose and the boosters. And in many others coverage, for example, the vaccine coverage for the young kids between 5 to 11, what is the impact in terms of for the entire population to achieve herd immunity. And our new focus is really building a new framework for integration of neural network and dynamic models, so that not only the model parameters can be estimated in real time, but also some of the mechanistic formulation can be informed from the real time, so that you can select the model and identify the parameters simultaneously and in real time. So within this lab, we also have a long-time relationship and collaboration with the Sanofi Pasteur, that's the largest vaccine provider in the world, but the largest vaccine provider in Canada. We work on different issues related to hospital infection, including AMR, and including the influenza, especially high-dose influenza in protecting the senior residents, and also childhood immunization program. Within COVID-19 and the collaboration between the academic community and the Sanofi has been focused on multiple issues, including the mass gathering. When you have mass gathering event like HOSH, like Winter Olympic, you have participants from different parts of the country, arriving and departing different time interval to bring different history and memory of the and vaccine products, and potentially in a event that could potentially be a super spreading event. And we're working on something related to the optimal dose intervals and also multivalent vaccine, potentially, for example, a COVID-19 and influenza vaccine combined. Maybe I should touch on a little bit on the most recently established national network called Mathematics for Public Health that's joint funded by our National Natural Science Foundation and by the Canadian Public Health Agency of Canada. The new network is led by myself and Professor Kumar Murthy of the Fields Institute on behalf of all the mathematical research institutes across Canada. We have interest in 4 different types of issues related to respiratory outbreaks, to vector-borne diseases, to foodborne outbreaks, and to AMR. So given a larger network, we are more focused on integrative way or integration, hybrid and complementary modeling framework and templates. And we're focused more on what we call systemic risk of pandemic and on using model to inform decision-making in the so-called emergency operations center. So we really want the model can communicate it to the highest level of command, but in a very timely, short duration decision-making process. Thank you very much.
Thank you, Dr. Wu. And this is amazing that you had the dialogue between the policymakers and you as an institution, which is sometimes is challenging to, and was it, I think we have more questions on that, but we can ask it during the question answer. Mac, if it is okay, we can start with a question to you, and maybe you can explain your research focus as well in the same thing. And this is in the interest of the time, actually, we are trying to speed up the process. Since this question that I'm thinking of asking you is on modeling base, so maybe Dr. Wu also can talk about something from his background on Ontario. So, but let me ask you, Mac, you this question, if it is okay with you. Is that fine? Okay, so Mac, I think you are muted, so what we have seen in this...
All right, so go ahead, Anish. So we have a dog in the room.
So what we have seen in this pandemic actually is wide variety of models coming up, especially in the starting. And there was conflicting conclusions in the sense that they have a different estimates for outbreak, for example. Mm-hmm. So how is the, and there has been criticism for one over another in the literature, in the news, for example. So from a modeling perspective or from the policymaker perspective, how do you choose which model to consider, which results to take in the prediction, for example? How do we decide? Because if we, our modelers, are creating those kind of confusion, of course, the policymakers are finding it much harder.
Okay, so let me go back on one of my favorite mantras here. So many of you know I'm also an artist as well as a mathematician. And Pablo Picasso once said, "Art is a lie that shows us the truth." And these models are lies that show us the truth. And that's never been more true than the current pandemic. Bach said that all models are wrong, but some are useful. These models, these lies can really mislead us. And I think that's, and the only, I'd say the only thing worse than having no model at all is having one where you don't understand the impact of the assumptions on the model. Because when this happens, you move ahead with false confidence. And all these models that give different predictions are based on different assumptions. And I think some of the fault also lies with our community. I've worked at Los Alamos for 40 years, and I started out as a physicist. And when you do a physics experiment, almost everyone seems to believe the results of the experiment except for the physicist that ran the experiment, because he knows all the caveats. And when I moved into the modeling epidemics, the world had flipped around. It seems that many of our epidemic modelers have far too much faith in their own models. And as you mentioned, the rest of the world, they're the one that are the skeptics now, because many times we've oversold what the models can do. Now, as modelers, it is our responsibility to know their limitations. And one way to do this, at least in painting, I use something called the principle of most bad. When I'm stuck on a painting and I don't know what to do next, I stand back and ask, what concerns me the most about this painting? And then I only focus on that little piece. And as modelers, this is what many of us have been doing. It's what I've been doing my whole career. We take our models and we ask what concerns us the most. And it might be heterogeneity and mixing in the population. Bus drivers mix far more than, say, someone in an old folks' home. Susceptibility, infectivity, mask wearing, not everyone is equally infectious. This epidemic has had more behavior changes in our population than anything we've tried to model before. Evolution of the pathogen, the Delta variant, the role of outliers, super spreaders, single people or events that are super spreaders have been crucial to the path of the epidemic. And so each model that takes, we can't include all of these in our models. This is too complex. So we focus on a few of these, and they do give different predictions. And what we really need to be far more conscious about is to relate our forecast to our underlying assumptions and our concerns about what is and isn't in the model. And until that, I think most of our models can be great for qualitative insights. But to give quantitative predictions, like we've seen a lot in the press, it's really hard for these mechanistic models to compete with, say, statistical models. But even the statistical models suffer from the assumption of stationarity. In other words, that the underlying phenomena are not going to change. Where through behavior change, Delta variants, mask wearing, there's a huge amount of variation in our underlying transmissibility within the population. So yes, we have predictions that are all over the map, and they're all based on different assumptions for the models. And we have been, I want to almost say, negligent in not making it clear on how these assumptions affect the predictions. That's a long-winded answer to your question, Anush, but it's something that's really probably the top priority for our community. Is that we have, man's got to, Clint Eastwood said it, a man's got to know his limitations. As modelers, we've got to know our limitations and express it to others.
So two things you touched quite in depth actually is the assumptions needs to be clearly mentioned, and also as a modeler you should know how much to sell your model for, like not to oversell it. Yes. And so decision makers can see the judgment call and differentiate between the model, if I'm summarizing in kind of nutshell what you're saying. So Dr. Wu, I want to go ahead.
Yeah, no, I just wanted to add that point where Dr. Hyman said about focusing, narrowing down the focus and understanding on the focus of the model. On what are we focusing? What is a problem that we are dealing with? And the assumptions, of course, that as you mentioned. Yeah, go ahead, Dr. Anuj.
Sure. Yeah, so just Dr. Wu, I want to touch base with you also on the same question, because you work with the Ontario Provincial Public Health Agency, and I think you probably know much better because you were directly dealing with these folks in public policy. How did they take as a model results? When you come out with a model result, were they comparing with the other results in the news and saying your model's results are little off or were on track, and which one to believe? What were their way of looking things, and what do you want to say on this?
Thank you for your question, and it's always good after Mark. His word is always has wisdom and experience. In terms of the province of Ontario, that's the province I'm serving and living in Canada. The public health decision making realized that at the early stage of the pandemic, they already realized their outcomes predictions from different modeling groups. They are not necessarily inconsistent with each other. So the government decided to commission what we call a modeling consensus table, where we bring all the relevant modeling groups together to cross-examine each other's forecasting before that forecast submitted to the science table and the decision making. So that's a table we share. The major difference between this pandemic and the past is really we have real-time access to the high-quality data in the province, and we share our assumptions, and then we cross-examine the outcomes. Of course, even with all of this, the outcome will be different. So we provide the government a cluster of outcomes for consideration. We always provide different scenarios, and since it's constant interaction between the decision-making body of the modeling table, so the possible scenario that need to be analyzed is limited because there are only few scenarios the government, the political decision body can take. So that's part of interaction is very helpful, and it's also helpful for the modeling and for the modelers to tell the decision making what kind of data is critical. And so they can direct on the surveillance data collection. So this is the first time that the data collection is directly informed by the modelers and it's directly used for decision making. I have to say that the consistency of modeling projections does not necessarily mean going to be a evidence to impose on to inform decision making. We have a very real experience in January of this year. 4 different modeling groups come with the same conclusion that told the government the epidemic curve they are seeing at that time in January, February is very deceptive. So we see case declining very quickly, but we already detect the mutant strain. So we told the government to choose two outbreaks, two epidemics going on, one is the wild strain, resident strain, one is the mutant strain, and the frequency of the mutant strain is increasing very quickly. So while you are seeing a transit decline by all cases, you are going to see a very large exponential explosion of the mutant strain. And because decision making is highly influenced by the behaviors of the public at that time, what is tired of locking down. So even though 4 models reach conclusive and consistent conclusion, that did not inform the decision. So it's a very complicated process. Yeah. But I think overall it is a process that the model has very good say about the interventions in the province of Ontario.
So you touched 2 very important points that you said about the validation. You did the validation of the models that gave the authenticity of your results, for example. And second thing, what you touched base also, the role of the model is not only for prediction, but also what data to collect in the field.
Yes.
And that is also you are kind of highlighting in your this is wonderful. I mean, I think your experience of this really helped us see how public policies are taking care of these modeling decisions. So thank you very much, Dr. Wu.
I think
Dr.
now we will
Birthwatch.
move to the health economist that we have
Yes,
in the
please.
panel. Dr. Kavitha, since you're an expert in health economics, we have this question for you. Any drug has a certain way of being administered, right? A certain dosage, certain frequency, not due to behavioral issues or illiteracy, ignorance of the patients, Sometimes what happens is drug overdosage or underdosage, it may happen. So in such scenarios, a new line of study opens up, like what are the effects of drug overdose? How do you study the behavior of a drug to newer circumstances which were not thought about before, right? And there are tremendous costs that are associated with improper drug intake. So are there models that link behavioral change to lifetime value of the treatment? Are there models existing? And if not, how do you use the previous models to incorporate these behavioral issues? Over to you, Dr. Kavitha.
Thank you for this question. So, as far as we are concerned, definitely due to this COVID, there is a paradigm shift for all the interventions. And yes, this question on drug adherence or drug overdose is really very important, and this is playing a major role as India is. It's not that people are not that literate, and they don't adhere to drug. We have seen this challenge in many diseases, starting from hypertension to tuberculosis, and we have come up with different models, different innovations for drug adherence. So as we were aware, for tuberculosis, patients are not taking proper course, they're not adhering to it, and it is becoming a resistant disease, which is again very difficult to cure. So again, on that, some innovations were there. A strip with which is digitally, some components are there with digital connected chips, so that healthcare personnel is informed that the patient is taking medicine or not. But yes, there is a huge scope, and models are there previously which were explaining this topic. But since this COVID is there, we really need to redesign and remodify those models to get a better picture. And in India, the problem, as our previous panelist, Dr. Hugh, was talking, for doing an analysis, we really need data, and on ground data, utilities, we really don't find. And then it is very difficult to design a model, to conclude a model with some concrete findings, which really can be applicable on ground. So that these are some challenges, but then yes, definitely we can do some modeling, and we can come up with some conclusions on this drug adherence part. What is the impact if drug overdose is there, or no, this drug adherence is missing? We can definitely come up with something. Thank you.
Thank you, Dr. Kavitha, for that wonderful answer to the question. Yeah, Anuj, Dr. Anuj.
Yeah, Dr. Kachu, very good point you made. What you highlighted is the data missing and the field level. So when we come down to models, we are just hypothesizing many of these things, especially like utilities you talked about. So I want to actually get the opinion of Dr. Ramachandran also on the same issue, because Dr. Ramachandran is talking about the treatments, drug development, and delivery aspects. And this aspect is very important to how do you deliver it when we have less empirical information on the ground level, or there is information but somewhere hidden. For examples, it's not really out in public. What can you say? I mean, the new treatment that you were talking about is coming in the about to come in the model, but do we have the data to model those things to efficacy of it in the long term, for example?
See, most of the small molecule treatments that have come out for COVID, if we are speaking for COVID, are not do not involve completely new molecules. Even if you take molnupiravir, it's a molecule which is originally from Ridgeback Biotherapeutics, and if I remember, from around 2003 onwards. The molecule which I am referring to, which we carried out clinical trials on, umifinovir, has been around for more than 20 years in Russia, China, and for example, you just mentioned the word Tamiflu, that is the oseltamivir. It's also an antiviral with limited efficacy in influenza with different mechanisms. But there has been a tendency, generally with regulators around the world, to be little more lax. Maybe rightly so in a pandemic, where you want to save life on the in the immediate front, versus looking at long-term side effects. But as we are now more and more into the pandemic with the availability of, say, vaccines, we have other treatments, monoclonal antibodies, and even better social adherence in many. And of course, we also have this thing about let's not follow COVID guidelines. This kind of the natural thing to say that throw everything to the wind. But then side effects are definitely very, very serious. And it's regulators who have to then effectively control the use of drugs as per the spread of the or the epidemiology and the spread of the infection in the population. So we have different levels. Emergency authorization. You would have read recently that the Covishield vaccine, the AstraZeneca thing equivalent in India itself, was previously authorized in an emergent manner, and recently they have approached the regulators for use in a regular manner. So it's an evolving situation. Uh, I'm sure Molnupiravir, when it comes, also will be under emergency authorization. Merck USA itself has applied to the regulators, FDA in the USA, only for high-risk patients and those who are very old. But it's too early now to develop modeling data. We really don't have proper therapy itself for the general population used in such a level. We do not have modeling data, but in a state like Uttar Pradesh, probably Dr. Brian Val would know. He's also involved in now strengthening the health response in Uttar Pradesh as one of his projects. I was seeing in his profile. The government took proactively all the steps that are necessary, all possible steps, as the pandemic evolved. It made those packets of ivermectin, vitamin C, and spread it in the population. Uh, it made the population more aware about the need to be to follow more COVID appropriate behavior, and the link between the micro response at the field to macro health policy initiatives. This could be more, I think, effectively established through the proactive approach of the government through health policies, etc. Even if you don't have a perfect response, it serves to alleviate the apprehension, the thing in the general populace, the panic, and then it comes to the next stages. These are some things that we have to think about, and as we go further along, and then probably this pandemic becomes an endemic situation, things will pan out, and we'll have more ex post facto kind of analysis rather than current
Yeah.
running analysis.
Well, I'm glad that because we as an outsider would have a difficult, as a modeler, has a difficult thing to know what is available and what is not. But coming out from you, actually, now we can see what is the challenge at the practical level. One thing we have seen in the treatment, for example, so suppose Tamiflu like COVID medicine come in the market, people like who are unvaccinated would prefer to go in that. They will change their potentially change their behavior, some of those ones, and they will take the medicine rather than taking a vaccine. So they would be probably more unvaccinated ideas propagating in the population because they think that the effective medicine is there.
I'll give you one very nice example. My student went to his village, and there he, as part of his project, he interviewed around 834 adults in the village on COVID behavior. And interestingly, if they found a person with a mask, there was a lot of social boycott because they believe the person who is wearing a mask must be having COVID, therefore he is wearing a mask. So there is hesitancy at all levels. Vaccine hesitancy is something, luckily, in the Indian context, we have less vaccine hesitancy, but it's the role of the more enlightened health service providers and people who are in the field to spread awareness. so that this link that even if you have a vaccine, there are effective breakthroughs which happen. Although you might have supposedly lesser number of or the severity of infection a little milder, but it's important, and this is where, as I mentioned below, the link between the micro therapeutic strategies and the macro health response has to be more strongly strengthened into alleviating the hesitancy in the population to, and also sensitization of the medical fraternity at large. It's surprising that quite a large bit of the medical fraternity also are quite ignorant about the latest treatment methodologies, which are more for the general population, and they also need to be sensitized a lot. This is another observation which I have come across while, as a researcher, looking at dealing with several clinicians.
Nice
Wonderful
observation.
example.
Yeah, go ahead, sir.
Go ahead. A wonderful example, I was saying, and I think as a mathematical modeler, we always have this behavior aspects. It's very challenging for us to incorporate. Dr. Kachu, you was about to say something.
Yeah. So that is what I wanted to, highlight here. Such models are existing in different countries, maybe, but we can't really come and replicate them as it is in our own country. As as the example was given by Professor Ravishankar, this villages in mass, they think it is something that they have COVID. So such components, there are 100 components in our country which we need to include, some we need to exclude. We can't really replicate the same model here in India. So it should be country specific with country specific components and data.
Thank you.
Very nice important point came out of this discussion. I would like to bring in kind attention. I see a lot of heterogeneity in terms of behavior, in terms of social status, and these are some things which drive the non-vaccinated population. They see lately there is a lot of localized COVID outbreak. This is because I feel unvaccinated people are not randomly distributed, but they're clusters of unvaccinated people. This is probably because of the political or geographical social divide. And there are a lot of reasons why we have these clusters where people are not getting vaccinated because of their financial status or because of decades of systematic racism that existed, which pushed some societies to the lower strata across the globe. So, what is the importance of this heterogeneity among people in models for understanding COVID interventions? And is this being incorporated in modeling, the heterogeneity part? This question is to Dr. Brian, because he's a public health expert. We just want to know whether this heterogeneity is being part of any of the models that are being used to study COVID interventions.
It's a fantastic question, and it's been a challenge, I know, for a number of different models. In the models that we have developed, for example, the integrating vaccine hesitancy has been a challenge, and for a couple of reasons. So one, I think in order to accurately kind of account for this kind of heterogeneity in a model, we of course need more data, better data to understand how these factors correlate, how they cluster, and therefore how they can be integrated into models effectively. I think also looking at novel approaches of integrating these factors into standard modeling approaches, or developing agent-based models that can handle these kind of behavioral approaches. I also kind of want to talk about, we've talked about heterogeneity within models, but I want to maybe talk about the heterogeneity of models themselves. And I think that there's also value in bringing together the heterogeneity of models and looking at ensemble models for thinking through the different kinds of questions that we want to answer through mathematical modeling. So the work that I presented earlier that was done and commissioned by the WHO, our group was one of several groups that aimed to answer key questions around in-person schooling, lifting of MPIs, minimizing the burden on, keeping health systems from exceeding certain thresholds, so on and so forth. So our models are being brought together with several other models to look at from an ensemble perspective what kind of reasonable approaches might make sense in for example, low and middle income countries, how that differs from other settings. So it's there have been a lot of mathematical models. The predictive mathematical models out of the US have been well captured in ensemble models that seem to relatively accurately, at least within a short time frame, maybe two or so weeks out, make predictions on the trajectory of the epidemic in the US. I have not seen any ensemble approaches to predictive models in India. I would love to see such an approach rather than just relying on one, two, or this model or that model or so on and so forth. The other issue in related to vaccination, where we see heterogeneity in India as well, that I think our models do not currently account for, but that may we may want to think about integrating those into our models. And it's across multiple states, the proportion of women who have been vaccinated appears to be, based on data provided by the government from the CoWIN app, seems to be less than males. And so that's one source of heterogeneity that we may also want to think about in terms of integrating that into different vaccination models that look at different vaccination strategies.
Yeah, this is amazing point that you talked about. We were talking about example as a vaccine, but I think you brought two other aspects of heterogeneity, like ensemble of model, which climate modeling field uses it quite heavily. But somehow the infectious disease dynamics field, I'm not sure if many people have used it.
In the COVID epidemic, it's the first time we've really seen that. If you look at some of the MIDAS and CDC websites for predictions, they don't just show one model, they show an ensemble. And then they show the median for the predictions, and then somehow get a standard error estimate from the range. But also what's often lost is this is it's a standard error. This is a standard error for the models, not a standard error for the actual prediction, the actual epidemic. But I want to go back to the point of, it's not just vaccines. One of the biggest caveats we have in our models is the distribution of susceptibility within the population. And this is not just true with COVID, it's also true with influenza, because we have an recurring epidemic. So we have susceptibility is greatly reduced by being vaccinated or previously having the disease. But there are also variations in susceptibility to infection due to just basic differences in our immune system, our age, the wearing of face mask, and social distancing. We have some communities where social distancing is observed and some where it's not. Those communities have very different susceptibility to infection in a single contact with another person. And when we want to incorporate these in the models, if we use a compartmental model, the underlying assumption of compartmental models that everyone in a compartment is indistinguishable. And so if we have a large variation in susceptibility to the vaccines or previous infections, then they can't be put in the same compartment if we expect an accurate prediction. So we either have to make a network of compartments or make the transition to an agent-based model, where every individual is assigned a level of susceptibility based on current behavior and maybe past infection or vaccines. So the models are, I think, rising to the challenge, but predicting epidemics is a lot messier and harder than predicting the weather. Weather is wonderful. It's based on fluid dynamics. And if you do an experiment with fluids, say water, and then you come back and do the same experiment with different water, you get the same answer. If you put a bunch of people in a room and see how they interact and write that down, and the next day you put a different group of people in the room and see how they interact, you're going to get a different answer. So we have to deal with the variability of people that doesn't exist in fluid dynamics. So we've got a much more difficult road ahead.
Yeah.
I
Thank you,
think
Mac.
the other.
Go ahead, Brad.
I would like to say one thing else for those that do fluid dynamics. One of the mantra of fluid dynamics is the butterfly effect and chaos, where we take big 3D, it was developed by Ed Lorenz based on weather equations. He came down to a system of 3 ordinary differential equations to kind of try to understand how weather behaves. And he showed how small changes in the initial conditions could cause large outputs. It's the beginning of chaos theory. No one would ever dream of predicting the weather using the system of 3 ODEs of Ed Lorenz. However, I do see people that are dreaming of predicting an epidemic using 3 ODEs called SIR. And they're about as relevant to an epidemic as the Lorenz equations are to predicting the weather. So we have to be careful about these kind of things.
Yeah. Go ahead, Brian, you were saying something?
No, I was just going to add that, another, and I saw this, play out as well in India. Models also don't exist in a vacuum. I mean, you could think of models as an intervention themselves as well, where behavior or policy or various other effects change as a result of models. And so I think it's important also in communicating around models, because you want to make sure that those caveats that go into a model, the assumptions are flexible to account for potential changes of behavior or changes of policy.
Thank you. Yeah. Dr. Wu, yeah, you are raising your hand, so go ahead.
It's just interesting to follow up with Mike and Brian's discussion. Just 2 days ago, I gave a talk in Interface Center in China, and tomorrow I'm going to give another conference about the big data. So one conference focused on what's next for challenge for the modeling, and the big data conference is really what is challenge and opportunity for big data. I saw they are all relevant to the issues about the complexity of modeling in the presence of heterogeneity and especially behavior changes. I do believe the need of big data and complexity of model is really because of heterogeneity, and one of the major driver of heterogeneity is the behaviors. And I think COVID-19 provides us a challenge, but also opportunity, because you can go back to locate the social media, locate the disease, to see how the behaviors changing over the course of pandemic. So those behavior data are more cultural, population specific, and that's coming to Dr. Kavitha's comments. We need those data. But those data can be collected, analyzed, and provide a data set for you to move ahead. Of course, it's going to change for different epidemic, for different pandemic. There might be different change behavior change, but the baseline is there. And this baseline can be used for you to quickly update your understanding of the behaviors, and so that you can move on. So this the biggest challenge for us really moving ahead is really when this one is in better shape, then we should really prepare for the future. And I think we did not learn a lesson from pandemic influenza. We think we did it well, and therefore we did not go back to see what happened. And this is the time I think we have to go back. And that will give you a lot of data for you to act forward.
This is amazing. I think, Dr. Bhadwaj, if you are okay, I think we are running short of the time, but the discussion is so interesting, and we are having more questions than the time permits us. I would like to probably get at this point, and I think Dr. Wu, you said it very well about the post-COVID. You were talking about post-COVID, what one should do, and maybe we can ask all the panelists a concluding remark on what do you think, where is we are heading in terms of modeling, for example, from your respective field. To Dr. Wu, to you first.
Thank you. I think we should tighten the reciprocal linkage between the decision-making process and the modeling process, and establish a link that during the peaceful time, so that during a wartime you can activate the linkage, so that you will accelerate the data transfer from the data collections to the modeling community. But you can also, again, direct the data community surveillance system what kind of data should be used. And if that linkage is there, then the decision maker may have more confidence in the cluster of models they are using to inform their decision making.
So your point is actually to strengthen the data collection process and make it available for both modelers and decision makers in a on real-time basis as much as possible.
Yes, and link them together. So through the data, you link the modeler to the decision maker together.
Okay. Dr. Ramachandran, what do you want to say? I'm going in a random order according to my screen. So please, Dr. Ramachandran.
Yeah. See, the panel discussion has severely highlighted that there are severe limitations in the modeling right now, primarily driven both by lack of data, access to good data, human behavior. It's like predicting the chaotic weather, butterfly fluttering its wings somewhere. But then, having said that, I think effective monitoring at the micro level is very critical, especially at this stage of the pandemic, when kind of it's all under relative control across the country and maybe in this thing, and we have to be prepared with tools to face the pandemic wherever it is. So here, things like what Dr. Kavitha Kachoru presented, the ability to deploy strategies at a relatively short time. And while we build up data to predict the emergence of infection pockets around resource-limited settings, I think this is very critical. Not only having the tools to fight the pandemic, but also to know effectively where to deploy those tools. Modeling will keep improving as we get more and more data as we get into the pandemic, and I think this will have a lasting impact, even in the ability to face future pandemics, which are anticipated now with the global warming changes and nascent viruses coming out, say from the Antarctic or the Arctic ice melting and so on and so forth. We are seeing such scary scenarios also.
So you talked about last leg. How do you deliver it practically to the people? This is amazing. Thank you, Dr. Kachoru. Floor is yours.
Okay, thank you so much. So I would really like to emphasize on the point, as you mentioned, Professor Ravi mentioned here, we need to collect data from the last mile. But in India, sir, that's really not possible, since we have been to field, how field staff is working, how they are entering the data. That's really not possible in the current scenario. But yes, there is a ray of hope, and there is a solution for it. And the solution is, as we're already moving towards digitization of everything, So if everything at the micro level is digitized, we can directly get the data, and that is what is a gold mine. That is what is a real-time data, which really we can use for any modeling, any design, any study, because it is directly capturing the real-time events which is happening at the field. One is that. Second is Indian indigenous data. Like we need to do more primary studies to build a capability and build a knowledge repository on Indian data in terms of quality, in terms of utilities, and in terms of everything and anything related to modeling, so that we have our own repository and we can deliver robust models which can really lead to some conclusions and that can be implemented across the country. So these are my closing remarks. Thank
This is
you.
wonderful. So you talk about regional strengthening, not only
Yes.
delivery and aspect, but regional strengthening of getting the data to the digital. That way, people can access it very, very effectively and quickly. Actually, a fantastic point. Thank you, Dr. Kachoru.
Sorry to interject here. Again, in a country like India, we are more or less, in terms of this modeling perspective, a collection of various countries. It's not just one country in terms of modeling,
True,
because
true,
the social
true.
response in different situations are different.
Well,
Very
for
true, sir.
example, supply chain comes from parts come from different countries. So that is a good example in this. So I will ask Dr. Brian Val your concluding remarks on.
So I'm going to, I think, echo what has already been said by the rest of the esteemed panel. I mean, your models are only going to be as strong as the data that go into them, and so ensuring that strong, reliable, believable data are being used in models is incredibly important. In the context of a limited resource setting during an epidemic, it's difficult, of course, to pursue efforts to collect data in parallel to response efforts. And so what I would say to that is that response efforts, as they are more and more digitized, there are opportunities as well through routine response efforts to make those data available for analysis, and those data can be fed into mathematical models as well. We've worked very closely with governments in Andhra Pradesh, Tamil Nadu, and have published a nice series of papers using routine surveillance data, surveillance data related to the response efforts over the last year and a half there, and have been working also with other states as well to do some analysis on the surveillance data that have been collected as a part of COVID. Of course, those have their own caveats that we need to be aware of and communicate around. But I think there's a nice opportunity, especially in India, where a lot of the response efforts have been digitized, to use those data and make those data available for epidemiologic analyses that can feed into models.
Thank you, Brian Val. Maybe we can ask now Dr. McHyman. You have been part of this MIDAS, which is a large network and of modelers. So what do you see as a post-COVID? Where are we heading, and is this sustainable? This type of explosion of models are sustainable?
So thanks for mentioning MIDAS. So about 20 years ago, the United States NIH realized that to give reliable answers to the questions we're asking about epidemics requires a sustained large-scale effort over decades with tens, if not hundreds, of modelers involved. They created a new community called MIDAS, which stands for Models for Infectious Disease Agent Study. Now, this organization, it's not, you might think of it as an organization like American Physics Society or Society of Industrial Applied Mathematics. It's a loosely organized group of individuals from around the world. It's open to the world modeling community. It's free to join. The goal of the organization is to develop and share better models for predicting epidemics, share data, and connect with the data collectors for the type of data that is needed to make the models more reliable. And also through Midas, they build strong connections with decision makers. There are regular meetings on Zoom, annual meetings before COVID or in person, where the modelers not only share ideas, but they bring in decision makers and panelists to understand what types of guidance the modelers can provide that would be useful and used in making decisions. We also critique each other's models, critique each other's data, ranging from simple compartmental ODE models to agent-based models, which now have been developed for 10 or 20 years that can handle millions of people, have tens of maybe as many as 100 programming years going into some of these models that have ended up costing $30, $40 million. So this is a community that's open to anyone that's involved in modeling or in policy decisions for epidemics that would like to engage. And so I think it's organizations like this that will be the glue that ties our community together. And I encourage people, just Google US NIH Midas, MIDAS, and there'll be a simple web form to fill out.
Well, thank you. You actually touch one important point, the critics of the models, and that is very important because the communication of the models have also exploded, and that has led scientists to communicate effectively in the media, actually social media or the news media. So, well, I'm very grateful to all of you, all the esteemed panelists here. Dr. Bhardwaj and I actually was looking forward very eagerly to this session. And it's amazing. We have so many questions that at this time, and we are getting list of questions.
Yeah, we have an overwhelming set of questions from the participants, but due to shortage of time, we are unable to ask these questions. So, sir, Dr. Anuj, your concluding remarks?
Sure. I mean, I just want to say thank you, all of you. We are very grateful for making this conference extremely good. Having been so many different disciplines coming and talking on a similar type of questions really helped modelers, because I am from a mathematics background. I know how people think from a mathematics discipline, and it has helped seeing the other side of the world as well. So I think I see Dr. Kachoru is raising her hand. So please go ahead.
Yes. Actually, since we are doing modeling, and in this panel, there are many esteemed, learned people who can help us in develop more robust models. So I would really request all of you to be open for collaboration or guidance so that we can develop good models, which can deliver some result.
Sure. I mean, I think we will do one thing, if you all are okay with it. We'll going to circulate the book of abstract with the contact information so that people can approach you all.
That would be really grateful.
Yeah.
Dr. Wu, you had a raised hand, or
no? I just want to sum up to
Thank you so much. Thank you so much, everyone.
Thank you very much,
everybody. I can see the cycle of this mathematical modeling, getting data, build model, and then prepare interventions, and then deploy interventions, get data again, optimize the model. I can see everybody involved in this process on the screen right now, and we pray to our Lord to bring fruits to our efforts in containing this pandemic as well as possible. Thank you all for participating in this fruitful discussion. Thank you.
Thank you so much.
Thank you.
Namaste.
Bye. Namaste.
Namaste.
Bye.
Thank you. Bye. I cannot hear you, Dr. Bhardwaj. You're saying something.
Dr. Anuj, can you hear me?
Yes. Now I can. Very slow. We cannot hear you. I cannot hear you.
Can you hear me now?
Yes.
Yeah. Thank you, Dr. Anuj, for such a
I'm sure all the participants have enjoyed this panel discussion. We will now have the next presentation, next keynote address. Dr. Anuj, I think Dr. Alun Lloyd is with us.
Sure.
I will briefly
Greetings,
introduce the
Alun.
next keynote speaker, Dr. Alun Lloyd. Dr. Alun is Drexel Professor of Mathematics and Director of the Biomathematics Graduate Program at the North Carolina State University. Following an undergraduate degree in mathematics from Cambridge University, he received a PhD in biological sciences from the University of Oxford under the supervision of Robert May. After postdocs at Oxford and the Institute of Advanced Study, Princeton, he took up a faculty position in the mathematics department at North Carolina State University. He has interests in infectious disease epidemiology with a particular interest in mosquito-borne infections. Dr. Alun, the floor is yours. Dr. Alun, please unmute yourself.
Okay, we should be good now.
Yeah, you're audible now. Thank you.
Very good. And do I have to do anything to make my slides be full screen? Can you see my slides?
Yes, we're able to see contact tracing for COVID.
Well, thank you very much for that introduction. It's a real pleasure to be here. I guess we get more used to remote talks by the day, but I'm still not fully used to them. I always prefer to be in person with the people I'm speaking to, but soon, hopefully. So I'm going to talk about a project that I was involved with looking at a control measure for COVID-19, so contact tracing. So back in the days before the vaccine, we were reliant on non-pharmaceutical interventions, many of which involved trying to isolate infectious people. So this project, this group was pulled together by Kevin Esveld, who's at MIT, the senior author of this work, but I was part of this group. So contact tracing has long been used in an attempt to control disease outbreaks. And the idea with typical you have a known case, you try to find and isolate people, contacts who that known case might have infected. And contact tracing is a difficult process. It's often difficult to identify, find, and get hold of all the contacts. It's often difficult to find these contacts quickly enough because obviously you've got to find them before they go on to cause secondary infections themselves. One question you might ask is whether you need to be sure whether someone is positive before you either trace from them or before you ask people to isolate themselves. And so, you know, we think about back in the early days of COVID, there were big issues with tracing and testing, rather. And one question that got asked early on is, can digital approaches help with this contact tracing? So traditionally, contact tracing involved, you find someone, you ask them about their contacts, you ask for information about those contacts, and then someone has to phone them up and get in touch with them. But the question is, can you make use of digital approaches? So for example, cell phones. Also, the fact that COVID presents many issues for contact tracing, and in particular about the timing of your ability to find contacts and to isolate them. So of course, as we know, we have pre-symptomatic transmission. So even if you have someone who is identified as a case, they may well have already infected people before they became a known case. Also, we have asymptomatic cases who wouldn't end up entering the contact tracing system, at least directly. So in this little cartoon here, we have the initial case in some area. This person happened to be asymptomatic, so we would never have identified them based on their symptoms, but they infected someone who became symptomatic. That person then entered the contact tracing system, and various outcomes happened. So, for example, some of their contacts might have been detected in time, they were isolated, they wouldn't cause secondary infections. It might be that one of the contacts was traced, but that tracing took too long, and by the time they were traced, they had caused secondary infections. And some of the contacts of that person might not have been able to be traced. So as I said, there was the suggestion that we could use cell phones to assist in identifying contacts, and particularly potentially risky contacts. So the idea here is that people have to be in proximity for some period of time in order for transmission to be likely. And with cell phones, either you can use the fact that cell phones have some notion of where they are, or that two cell phones can figure out, for example, by using Bluetooth communication, whether two cell phones are close to each other. So you can potentially identify contacts using cell phones. And the one thing you can say, and this is has been a big issue, is that these sort of cell phone-based approaches raise significant privacy issues. So, if your phone is keeping track of who you're in contact with, that's a lot of information that maybe you wouldn't like to be out there available to someone. And so there are significant privacy issues that raised by people, but were also raised by the cell phone companies. So Apple and Google instituted COVID contact tracing protocols within their phones, but they were quite careful that the data was basically kept locally on a phone rather than in a centralized database. So our proposal was what we call bi-directional contact tracing, which is not only trying to look forward to figure out who a case infected, but looking backwards in time to try to figure out who infected the known case and to continue tracing from there. And operationally, that means that you need to know about contacts much longer period back from the date when symptoms started being shown, compared to what were at the time the current guidelines for contact tracing. And of course, because you're looking over a longer period of time, you're going to pick up more contacts. And so it involves a lot more work, but it turns out it can significantly enhance the effectiveness of contact tracing. So in that picture we had before, where we had the known case, if we can also look backwards in time, not only forwards in time, we might have found the person who infected our known case and then be able to trace outwards from that person as well. So we would catch more of the transmission events in our contact tracing dragnet. So in order to study the effectiveness of contact tracing, we used a branching process model. So this is a very simple model of how transmission works. And this is a model that has a long history of use in the literature for studying contact tracing. And even in the COVID literature, there was a prominent paper back in the beginning of 2020, Helliwell et al., from the London School of Hygiene and Tropical Medicine, used this branching process model they used it to look at forward contact tracing, but we adapted this model to account for the possibility of bidirectional tracing. And so the one thing that many of these studies of contact tracing have found, and as I've already said, is that the timing of different events is crucial. So the timing of the life history of infection, things like onset of symptoms, when does infectiousness start, and then the timing in the contact tracing system, so delays in the tracing, in being able to identify contacts, the time taken to test and figure out if someone is actually positive or not, if that's needed, which it is, because as we know, many people are asymptomatic, so you really want to test people and not just rely on symptoms. And then the time taken to relay results back and for people to start isolating. So all this timing of all these different events is absolutely crucial in terms of the effectiveness or potential effectiveness of contact tracing. And so this timing, these distributions of time intervals, for example, from someone getting infected to their symptoms, the timing between someone being infected and causing secondary infections, the timing between someone being symptomatic and entering the contact tracing system, so on, all these things are accounted for in this branching process model. And we have a whole table of model parameters. A couple of things to mention. In common with some other studies, we assume that there is some percentage of transmission that happens environmentally rather than by direct individual-to-individual contact. The extent to which that happens could be debated, but this will be an absolutely crucial thing in terms of the ability of contact tracing to control an outbreak. So we have to make somewhat conservative assumptions on that. The other thing we allow for is an over-dispersed distribution of secondary infections, reflecting sort of super spreading or differences in levels of viral shedding across a population. And we took, based on some other studies, a negative binomial distribution with this K parameter to be 0.11. So that represents a fairly over-dispersed distribution of secondary infections between different individuals, accounting for the potential of super spreading in the population. So we can look at whether the standard sort of contact tracing approach would control COVID-19. And because we were modeling this back in the spring of 2020, we took a basic reproduction number of 2.5, the wild type. And we can look at, for example, how the effective reproduction number depends on the fraction of contacts that are able to be traced. And also, we start off our simulations with 20 introduced infected individuals. We imagine we haven't spotted the very first case in a given population. And we ask, what's the probability that we're going to control the outbreak using contact tracing? And the message is the standard approach at the time for doing contact tracing would be unable to control an infection that has a basic reproduction number of 2.5. But we find that we can control, if we add in bidirectional tracing to this protocol, we find that we can increase the percentage of outbreaks that are able to be controlled and cause further decreases in the effective reproduction number. So we can bring that effective reproduction number down close towards 1 if we have a high probability of trace success and we do bidirectional tracing in which we retain information on contacts for 7 days. So we were thinking also, as I said, about the use of cell phones to achieve this, so this so-called digital contact tracing. And if you think about it, so not all of the population is going to have cell phones that are able to do this, to participate in this contact tracing. So you have to have a smartphone, and you have to have downloaded an app. And then, even then, not everyone who has the ability to do this will buy into the system. So not everyone will be willing to share their contact data. And what we found was that the performance of the digital contact tracing is highly sensitive to the fraction of people who have the smartphones that can run the system, and then the fraction of those people who are prepared to participate in the system. And that really this digital system is only going to be effective if those uptake rates are really quite high. And one of the reasons for that is that if you imagine, in order to sort of identify or to remove a potentially risky contact, the person who has the infection has to be in the system, and the potentially risky contact has to be in the system. So there's a quadratic dependence there on the uptake rate. But we found that if you combine both the conventional contact tracing and the digital contact tracing, you have a higher likelihood of succeeding. So the idea being that the digital contact tracing takes you so far, and then the manual contact tracing acts on top. So you can basically synergize between the two approaches. Much higher probability of controlling an outbreak and a much larger reduction in the effective reproduction number. So we're getting this effective reproduction number being pushed down closer to 1. Again, this is looking at that in a slightly different way. Again, here is a function of the probability that if we have a contact, we identify the contact. Incidentally, you might wonder here, why even if we have 0% probability of tracing contacts, do we have a reduction in the reproduction number below the baseline of 2.5? And that's because simply by identifying cases in this model, we're isolating those cases. So even though we can't identify contacts of those cases when this trace probability is 0, the fact that we're identifying cases and doing some isolation of cases has a modest impact on the basic reproduction number. And so here we see that the percentage of outbreaks controlled can be, reasonable if we combine manual and digital contact tracing, and the probability of tracing a contact is reasonably high. Of course, if the probability of tracing a contact is low, contact tracing is never going to be effective, whatever you do. So I mentioned before the fact that in our branching process model, we have heterogeneity in the numbers of secondary infections. And so this heterogeneity in contacts, and hence, one way in which that heterogeneity in secondary infections can arise is because different people have different numbers of contacts. And so in a social network where individuals have differing levels of activity, highly connected individuals have more contacts. And so if you look at a randomly chosen contact of a given person, they're more likely to be a highly connected individual. So contact tracing naturally tends to find highly connected individuals. And this fact actually has been known for a long time. This is a sociology paper whose title I like, but it also makes me feel rather sad. Why your friends have more friends than you do. It exactly reflects this fact that if you choose contacts at random from a population where where different people have different numbers of contacts, the people who have more contacts are more likely to be represented in the population making contacts. And so if you choose contacts at random, those contacts are more likely to be with a highly connected individual. And so this is one of the reasons why this bidirectional contact tracing, or this is related, this heterogeneity is why reverse contact tracing can be more effective. So if you have a situation where most people have few secondary infections, while a few have many secondary infections, so if we have super spreaders or super shedders, when you trace forward, you will often find a typical individual in the population. Those typical individuals have few secondary infections. But because super shedders or super spreaders cause lots of infections, a given person's infection is more likely to have been caused by one of those people than by a typical individual. So if you find the people who infect people, they typically are the ones who cause more secondary infections than a typical individual. So looking backwards in time, you're more likely to find one of these super shedders or super spreaders than a typical individual. And so backward tracing is going to be more effective than forward contact tracing. And in fact, there are some situations, some settings in which contact tracing has been rather successful. In places like Singapore, Japan, and even in some outbreaks in the state of Washington, where the contact tracing approach either explicitly or implicitly allowed for backwards contact tracing, or people did what we call cluster analysis, which has the aim of identifying the people who infected people. So one thing I want to mention is our approach used a branching process model, which only models transmission events. And the other approach you could use, or another approach you could use, is to have a full individual-based or a network model of a population. So as I said, this branching process is a commonly used framework in this setting for evaluating contact tracing and other non-pharmaceutical interventions. It's really quite amenable to analysis, so you can do R0 calculations. But as I said, the issue with this branching process model is that it only talks about transmission events that happen. So there was a picture of a tree I showed you earlier on, a transmission tree, and it just considers the successful transmission events rather than keeping track of all the contacts. And so that's actually quite important for contact tracing, in the sense that one of the issues with contact tracing is that you identify people who've possibly been infected, but maybe haven't been infected, and you ask those people to isolate themselves. So contact tracing can have lots of what you might think of as being collateral damage in terms of, for example, economic activity. If you ask lots of people to isolate, they can't go to work. You have to, kids can't go to schools and so on. So there is a downside to identifying contacts who you are not sure are actually infectious. Another issue, one big issue with the branching process model, it fails to capture many aspects of social structure. So we know, for example, that households, workplaces, and schools are important locations for transmission. We know that real-world population has lots of local spatial structure and overlaps in contact networks of individuals. So there are cliques. We know that individuals have repeated contacts with the same individuals. Lots of this sort of structure is completely missing from the branching process description. So in many ways, the branching process likely represents a worst-case scenario in terms of controlling infection. On the other hand, the branching process setup is fairly simple, straightforward, makes fewer assumptions, and is much more transparent in its formulation. One of the big problems with a full individual-based network model is that it's often very difficult to convey all of the ingredients, all of the details, and be sure that you've got all of those right in the network model, and have a full appreciation of how those assumptions impact the predictions you make. So I just want to say there was a paper in Nature last spring that talked about, so this contact tracing, actually using digital approach, had been deployed in a major way in the UK. So the National Health Service deployed a COVID-19 app that came in for lots of criticism at various times during its somewhat troubled development. But later on, that app came used by quite a large fraction of the population, in part because various venues would say that you can only, they would encourage participation, saying, you can come into our pub if you are registered with the app. And these guys from Oxford who were involved with the development and analysis of the data they were involved with the development of the system and analysis of the data, they claim that based on model predictions, that between 100,000 and 900,000 cases of COVID-19 were averted in the UK. Curiously, a majority of these cases were averted during just a 2-month period. And that 2-month period came after there was an upgrade to the algorithm that determined contacts. And that algorithm change actually doubled the number of notifications. So to summarize, the fact that COVID-19 has this sort of particular life cycle, natural history, where we have both pre-symptomatic transmission and the fact that we have a large number of asymptomatic infections who cause transmission events, this makes control by contact tracing very challenging for COVID-19. Our analysis showed that suggests that digital approaches are not a silver bullet unless there is near universal adoption and use. Although I should say the Oxford analysis mentioned on the previous slide said that digital approaches had a much larger impact. And one of the reasons possibly is the fact that they used this full, much more detailed individual-based model that had much more realistic description of social structure than the branching process model used. Remember I said the branching process is very much a worst-case scenario. Anecdotally, the bidirectional approach, we said the bidirectional approach can greatly increase the effectiveness of contact tracing, and anecdotally, this might well explain the success of contact tracing in some settings. And of course, I should say our analysis here was just considering the use of contact tracing and isolation to control spread. Of course, there were other mitigation methods going on, such as social distancing, mask wearing, and so on. And so we made the point that other approaches, even if they're only mildly effective, are going to be synergistic and will help bring that effective reproduction number below 1. And many more details of this work appear in the paper by the paper that came out of this work, Bradshaw et al., which appeared in Nature Communications in the start of this year. And I think I'll finish there.
Hello, Dr. Lloyd. How are you doing?
Yes,
Greetings.
thank you.
Well, first of all, maybe Dr. Vamsi, we can ask a audience question, but I want to say one thing. What you said is the first line about more interactive. The online is about. I mean, you as a speaker were not seeing other people, but we were listening it very carefully. So I just want to assure you about that.
No,
So.
no, the thing I don't like about online is that you can't see people. You often can't. It's often difficult to see people as you're talking. And so
Right.
I like to be in front of an audience where I can see if someone is puzzled or if there's something that they didn't understand or something they liked. I enjoy that feedback, and I miss that enormously. I'm looking forward to getting it again.
Sure.
Dr. We are also actually, not any questions from audience. We are also eagerly looking forward for the next this series, whatever we hold. Maybe physically we can have all of you here. That's in line. So, Dr. Anish, do you have any questions to?
Yeah, actually, I want to point out one thing that some of the audience might see it an interesting point here. What Dr. Lloyd's research is talking about is, of course, the digital data is one thing, and we are now talking about lots of digital data, social media data, apps data. This is an apps data that he was mentioning about contact tracing. There's different types of data, geographical satellite data, for example. So now the question is, when we use this digital data, there is a need for validation with the real life data. And that is what you have talked about today, actually, that you are comparing that digital efficacy or impact from the original data. And,
Yeah, I think, early in the epidemic, there were, letters in very prominent journals basically saying that, digital contact tracing or location-based information that came from cell phones would be a game changer in terms of controlling these infections. Now, that didn't happen. In many settings, that didn't happen. And so, I think it's one of our jobs as modelers. It's easy to make these claims, but we have to always, evaluate those claims and look critically at those claims. Now, the UK folks said that digital contact tracing helped quite a lot. But I noticed, as I said, that was kind of late in the day that it helped out, and that process, that digital system had quite a long and troubled rollout period. Mm-hmm. Of course, maybe next time, heaven help us, next time we will be better prepared. But I agree with the point that the digital data has to be validated. So we've seen lots of things like using Google searches, and so Google search trends, that sort of thing. The websites where people self-report symptoms, for instance, so you can get an idea. And those are great data streams, but as you say, they really require critical evaluation and validation before, so that we know how much trust we put in them, how much should we invest in those systems, what are the best ways that we can do things. And as I say, I also mentioned this business of collateral damage. It's something that we didn't think about much because we were just doing a modeling study. Our modeling study didn't take into account what is the capacity of people to do contact tracing. And so, it's very easy for us to say, oh, you should ask about contact 7 days back rather than just 2 days back. But that increases the number of contacts you have to look at. That increases the workload of contact tracing enormously. And that that is very difficult to scale when you have people doing the work. The digital system offers you the opportunity to scale it more easily. But then if there are issues with the digital system, what's the benefit?
So you think you actually touched the base a little bit in your talk also. The one of the issue probably is an ethical issues of the use of this.
Yes.
And if there is a modeling world that could help in that ethical issues, probably not. I'm just throwing a random question on that.
No. So as I said, one of the big debates was whether the data on contacts should be kept by the phone, and then if you are a case, then your phone will say who should be contacted, basically, or whether all of that information should be centralized, whether a single server should have all the information on contacts. Now, of course, from a modeler's viewpoint, I would love to see information on all of the contacts of population. But of course, you might not I might not want you to know all the people who I've interacted with in the last 7 days. I can imagine some scenarios in which I might not want to share that information. So I think there are, that is really not a modeling question, but there are certainly sort of, computer science and questions to do with how do you protect data? How do you do data privacy? What can be shared safely? What can't be shared? We've seen this with, the last few weeks, the last few years have shown us the dangers of social networks in terms of, how social network information can be applied, not always for the best uses.
For example,
question. Yes, sir.
Go ahead, Dr. Vamsi.
I have a quick question, Professor Alun.
Yeah.
At this point of time, where the COVID seems to be little under control, the virus.
Yeah.
So what I wanted to ask was, how at this point of time does contact tracing research at this point of time? So what is the stand? What is the status, or what is the stance of contact tracing?
That's a very good point, and it's one I didn't really emphasize enough. Contact tracing is most useful early on in an outbreak.
Yes.
Because if you have lots of transmission, your contact tracing system is going to be swamped. So contact tracing at this point could be helpful if you get like an explosive local outbreak. It could be useful in trying to keep a lid on local outbreaks. We also published a follow-up that said that contact tracing could be useful in terms of mitigating the spread of variants. So it's early on, and of course, there are some places where they have sort of managed to suppress local transmission, and then contact tracing will be helpful if there is an introduced case that could lead to an outbreak. But clearly, at this point, we have vaccines. And
vaccine and medication is also well established for COVID.
So just on that same lines, probably if we look it into a different way, the contact tracing methods could also be used in tracking unvaccinated clusters. And probably there are clusters of these unvaccinated localized clusters to identify and directing the interventions in that way. What do you think?
I agree.
So, sure. So as there are no questions, let us all thank Professor Alun Lloyd for his interesting and insightful talk. Thank you, Professor Alun Lloyd from the organizing committee, both me and Dr. Anuj. So thank you, participants, for your active participation throughout this evening session. So we will close the day, and tomorrow morning we will assemble we will start the virtual session at 9 a.m. IST sharp. Thank you. Sai Ram.
Helen, thank you very much for being part of it. It really means a lot to me. And
you're more than welcome.
Yeah, you are always such a supporter, and whenever I approach you for something, you have always been very helpful. So thank you very much. I really.
So where are you right now? Are you?
Yeah, so I'm visiting my parents nowadays. So I am in
India.
I
saw that you were visiting. I wasn't sure if you were still visiting.
Yeah, I am still. I will be here. So this is the time is like usually you cannot travel because of the semester. The semester you are teaching, and this this time is the festive season. Actually, there are lots of festival at the same time. So I did not.
Yes.
Yeah. So it was a blessing in disguise for like COVID outbreak, basically.